Right. So this is monocot leaf internal structure. The last class we completed dicot leaf. Let me take a pen. Right. So if you take a leaf internal structure, same as dicot leaf. In dicot leaf, mainly the three regions: epidermis, mesophyll cells, and vascular bundle. Here also same: epidermis, mesophyll cells, and vascular bundle. And the small difference between monocot leaf and dicot leaf. While I explain the monocot leaf, just compare with dicot leaf also. Right. First, in terms of epidermis, the both in both, that means dicot and monocot, have the epidermis, that is upper epidermis. Here you can see upper epidermis, upper side, as well as lower epidermis. So it is separated into two types, like dicot leaf epidermis. Right. In both epidermal leaves without intercellular space, dicot leaf without intercellular space, uniseriate, and monocot leaf also without intercellular space and uniseriate. But very few, some few leaves have the multiseriate. For example, in dicot nerea multiseriate, but in monocot phonix multiseriate. So there are the rare cases, right? So in both cases, it is covered by the cuticle layer. Cuticle layer present in dicot leaf and cuticle layer present in monocot leaf. But here some monocots, that means waxy leaf, the small difference, cutinization in dicot, but in monocot some leaves cuticularization, cuticularization, cuticularization. That means here the difference between the cutin in dicot leaf, cutin formation cell wall itself, not above, not surface the cell wall. The cell wall itself, the cutin present. It is impregnant with the cell wall layers. Whereas in monocot leaves, the cuticle layer above the cell wall. The cuticle layer above the cell wall, and it is appear as a waxy. And you can compare with some monocot leaf do not wet because of the presence of cuticularization. When rain occurs in dicot leaf, they can wet, but monocot leaves do not wet because of cuticularization. That is the another difference between dicot leaf and monocot leaf. I am telling you again, but not all monocot leaf. Some monocot leaf, I am telling. Right. So another important difference between in terms of epidermal cells, the special cells are present in upper epidermal cells. They are present only in monocot cells, but not in dicot cell. The specialized upper epidermal cells called as bulliform cells or motor cells. These enlarged cells. This is, you can see, this is upper epidermis. They are all equal in size, but whereas these two B for B, bulliform or motor, B. I have written B. These are called bulliform cells, are not given here. You can write here, or motor cells, called as motor cells. So motor cell function unrolling the leaf. When the wind velocity high, automatically the leaf unrolling. Sorry, the leaves rolling to prevent the loss of transpiration. So that is the importance of bulliform cells or motor cells. They are located at upper epidermis, but they are not present in dicot leaf. Remember it. So this is about the epidermis. And next, coming to the mesophyll cells. The rest of the place, this is all together. This is called mesophyll cells. This is the empty, the middle. Except this pink color, except the pink round shaped structure, that these are called vascular bundle. Except the vascular bundle, the rest of the place called as mesophyll cell. But in dicot, the mesophyll cells classified into two clear distinct region. such as polysaid cells and spongy cells. But in monocot, there is no separation within the mesophyll cells. All mesophyll cells in the form of spongy or polysaid and less internal space when compared with dicot internal leaf. But dicot internal leaf, especially in spongy cells, more internal space. But here the less internal space, and the both mesophyll cells have the chloroplast. Both consist of chloroplast. But one difference, that is mesophils does not differentiated into sponge and polysaid. But in dicot, clearly separated as polysaid and sponge. That is the another difference between dicot and monocot leaf. And next one, vascular bundle. First of all, the arrangement of vascular bundle. uh, just wait, my voice is clear or not? Right. In vascular bundle, first the vascular bundles randomly arranged in dicot leaf, but in monocot leaf, the vascular bundles almost parallel arrangement. And most of the and in dicot leaf, the vascular bundles dimorphic. That means the small vascular bundle, large vascular bundle are there. But here almost similar type of vascular bundle are present in monocot leaves, but very rare. They also have the dimorphic vascular bundle. So this is in terms of vascular bundle. But one important, the arrangement, random arrangement dicot leaf, parallel arrangement in monocot leaf. Right. And this vascular bundle surrounded by sheath, that is called bundle sheath. You can see here. You can see this is bundle sheath. The same bundle sheath also present in dicot leaf also. This bundle sheath made up of parenchyma cells in both dicot and monocot, but in larger, of course, almost monocot leaf large vascular bundle rather than small. But here, of course, here given the small vascular bundle also, but most of the monocot leaf only one type of vascular bundle, that is the large vascular bundle. And of course, it is sheath developed by the parenchymatous, but extended by sclerenchyma cells. This is sclerenchyma, this one. But this sclerenchyma absent in dicot. This is sclerenchyma, but this is absent in dicot. That is important. And this is protoxylem here. It is upper side, and remember one thing, and in vascular bundle towards the upper epidermis xylem, and towards the lower epidermis lower side, this is the phloem. And this arrangement same as dicot leaf. But in dicot leaf, we cannot recognize it as the where is protoxylem, where is metaxylem in normal situation. But in monocot leaf, we can identify as the protoxylem. This is protoxylem. Clear protoxylem present. But in dicot leaf, there is no clear protoxylem. And it forms the lacuna. It's called as lacuna. And this is these two metaxylems, the above. So these are the called V-shaped arrangement, clear distinguish. Another important in bundle sheath. In bundle sheath of monocot leaf, you have the chloroplast. Let me change color. You have the chloroplast. Here I have the chloroplast. Of course, chloroplast present in mesophyll cells as well as in bundle sheath cells. But bundle sheath cells, chloroplast present but does not form the grana. That is another important. So, but in monocot leaf, the carbon fixation takes place inside the bundle sheath cell. Received mesophyll cells, but fixation in bundle sheath cell. So these are the bundle sheath cell chloroplast. but the bundle sheath does not have the chloroplast in dicot leaf. This is the difference between dicot leaf and monocot leaf. And in our book also clearly given the differences and diagram, and clear clear diagram given, and clearly give the difference also. After the completion of dicot and monocot, you should see the differences. This is very, very important. If you understand differences between dicot leaf and monocot leaf, you can easily understand the both content, right? The page number 43 clearly given about the dicot leaf and monocot leaf differences. That is very, very important, remember. And sorry. Right, next one, leaf, sorry, root. Root internal structure, next leaf internal structure, sorry, stem internal structure. Let me stop this one. Right. So let's see small differences between dicot leaf and monocot leaf. Already told you, but remember me. Dicot leaf, that is dorsiventral, monocot leaf isobilateral. Upper surface dark, but both the surface in same color. And I forgot one thing: the silica deposition present in monocot leaf. The silica deposition absent in dicot leaf. And the arrangement of the stomata in dicot leaf only high at the upper epidermis, but in monocot they are uniformly arranged. And Godson's kidney shape in dicot, God shape, dumbbell shape. Gods are dumbbell shaped in monocot. And mesophil heterogeneous, such as spongy and polysaid. But in monocot, they are homogeneous, only spongy cells. And next, the vein is reticulate venation, whereas in monocot, parallel venation. Already told you protoxylem not distinguishable, but here the protoxylem distinguishable. Bundle sheath is single layer. Bundle sheath is one or multi-layered. Maybe one or multi-layer. Bundle sheath lack chloroplast in dicot. Bundle sheath consisting chloroplast. Bundle sheath extension is parenchymatous, but bundle sheath extension sclerenchymatous in monocot. So these are and the bundles are dimorphic, smaller and larger. But in the monocot, mostly the bundles are monomorphic. They are the larger one. And any other differences? Right. So this is about the dicot leaf. Now, so let's see. The epidermis, this is the first let me complete in dicot root. Dicot root. Already told you, I did not notice that my audio in off. Now it is on. Dicot root, monocot root difference. If the both dicot and monocot clear three region internally, one is epidermis, second one is cortex, third one is steel. This empty place cortex and this middle place called as steel. Now let's discuss one by one. The epidermis outermost layer like a leaf, and this is similar in stem also further. A single layer uniciliate intercellars without intercellular space, and it is covered by cuticularized, and which prevents the loss of water. And the very rare stomata are present, very rare, that is very rare. So this is about the dicot stomata. Whereas in monocot, monocot root stomata, if you see the monocot root, sorry, primary, oh, just wait, root. One minute. Right, actually, right. Right. If you see the monocot root, here also same epidermis, cortex, and endodermis. The epidermis, this outermost layer, the cells are closely packed without intercellular space. Same, the cuticle and stomata are absent. Uh, Here actually in dicot root also, cuticle and stomata both are absent. The cuticle and stomata both are present in leaf and stem only. But whereas in root, no cuticle and no stomata, right? Whereas in monocot root also no cuticle and no stomata. And the from the epidermis, the root hairs are erased in both dicot. Dicot also from the epidermis, root hairs erased out. In monocot also root hairs erased out. The function of root hairs absorption of water. So this is about epidermis. Almost similar, almost similar in dicot root and monocot roots in terms of epidermis. And the cortex. If you see the cortex, this empty place cortex region, here the cortex multi-layered. You can see here. Of course, this is a first layer. Outer layer is epidermis. The remaining layers, where what you see here, they are all belongs to cortex cells. So the cortexal multi-layer region made up of thin-walled cells, and this passes these cells also no chloroplast but may contain leucoplast. Here the cell contain leucoplast. They are the white colorless plastids. The function of these leucoplasts, the storage grains, storage food material, and and if you see the same cortex in the monocot root, same, no changes. The number of cells without uh, sorry, with some small intercellular space for exchange gases, and also it is also consisting leucoplast and also store the food material. So there are no changes in the cortex region also in both. And the so in the last part of the cortex, endodermis. This is the innermost layer of the cortex, cover the steel. It consists the compact cells without intercellular space like a epidermis. Most of the cells are characterized by the presence of special thickening, suberin. These are called transparent strips. The endodermis cells near opposite to protoxylem have no casparian strip because, for example, here epidermis, uh, let's change color, blue color cortex layer. The last cortex layer is the endodermis. This is endodermis. Of course, this endodermis, let's change color. Thickening, have the thickening, red colors, thickening, like thickenings are there. These are called Casparian bonds. But these Casparian bonds, they leave the gaps where the exactly protoxylem. This, for example, this is the continuation, this layer, this region, steel. In steel, this is xylem. This is xylem. Where the xylem present, exact opposite, there is no thickenings. There is no Casparian bond. This is called passage cells. The function of passage cells, for example, this epidermis, from epidermis, rootlet, from rootlet, if rootlet, if observe the water, this water trans movement from the epidermal cells to cortex cell, cortex to endodermis. But as it consisting endodermis Casparian bond, this Casparian bonds not allow the water inside. This water prevent. That's why the small some cells have the passage cells in endodermis that allow the water into the xylem cells. From xylem, root xylem, it continues to the shoot xylem and continues to the leaf xylem and transpirate out. So this is the function of passage cells. Remember, this passage cell present in endodermal cells. The same way in monocot endodermis also same. This is the inner boundary cortex which is unilayered like dicot root made up of barrel cell, intercellular without intercellular space. The young cells possess an internal strip, suberin. Same here also suberin, phosphorin strip. Due to additional thickening, this soon becomes a indistinguishable. The cells that lie opposite to protoxylem remain unthickening. Same, that is called passage cells or transform cells. No changes in terms of endodermis in dicot root, monocot roots, remember. And pericycle. So pericycle, this is the pericycle. This is the first part of the steel. This first part, this is steel. So first part of the steel called pericycle. In monocot root also same. Monocot root also same. First cycle, pericycle. First layer. The outermost layer of the steel composed uniseriate, maybe sometime multiseriate, and this is made up of parenchyma. The same like epidermis, endodermis, it is also arranged without intercellular spaces. And this pericycle absent in some hydrophytes and dicots. Some hydrophytic root and dicots. Why? Because actually from the pericycle, lateral roots are developed. What is the function of lateral roots? These lateral roots much branched and penetrate into the soil and absorb the water particles. But whereas in water particles, there is no point to penetrate in water. That's why most of the water hydrophytic plants, there is no pericycle absent. And the lateral roots endogenes, I already told you that during secondary growth, pericycle produce secondary cambium and phloem. I will explain clearly in secondary growth in further class. And here the pericycle, say in monocot root also pericycle outermost, the steel, below the epidermis, uniseriate or multiseriate. But like dicot root pericycle, monocot root cycle does not form cambium, does not involved in secondary growth. That is one difference. In dicot pericycle shows the secondary growth, but monocot root does not show. And the main variation only in steel vascular bundle. First, let's see dicot root. Vascular bundle, almost pericycle completed, and remaining vascular bundle and pith. That means if you take the steel, if take the steel, three regions present inside the steel. One is pericycle completed, pericycle. One is pericycle that is completed. Second one is vascular bundle not completed. I will explain now vascular bundle. And the third one medulla or pith. Medulla or pith. Right. So in steel region, one is completed. Pericycle completed in both dicot and monocot. Now the vascular bundle. The vascular bundle number 2 to 8, 1, 2 to dia condition. These are separated, alternate, radial, exarch, and arranged in the form of ring. So the arrangement radial, exarch, and maybe, for example, in this diagram, this is a tetrarch condition. That means the number of vascular bundle 2 to 8. Already told you vascular bundle number maybe that is the different species 2 to 8. But in diagram we can see the 4, 1, 2, 3, 4 tetrarch condition. So anyway, the arrangement radial arrangement. Radial arrangement means this is phloem. This is P for phloem, and this is xylem. This round zeros like structure xylem, and again phloem. Again xylem, again phloem, this portion phloem, and again xylem, and again phloem, right? So this type of arrangement there, and the arrangement of xylem also outside. That means protoxylem outside, metaxylem inside. So this condition called as exarch condition. That means exarch condition. Let me draw clearly here. Almost medulla may be less or disappear. We can't see the medulla here. So let me draw here this arrangement actually. First exarch. This type of arrangement. Here, this type of arrangement, this xylem arrangement, this type. Exarch condition. So how many exarch here? 1, 2, 3, 4. So this is called tetrarch. If only 2, that is called diarch. So this type of arrangement present. If you see the vascular bundles in monocots, so the number of polyarch, that is more than 8 polyarch condition. You can see here condition also. Here only 4. Here number 1, 2, 3, 4, 5, 6 number polyarch condition. This is xylem as it is, and center position phloem, and sorry, center position phloem. This is xylem, and this is phloem, xylem, phloem, xylem, phloem. This empty place phloem, remember, that's it. And same condition here also. A vascular bundle type is same. The type of vascular bundle uh, that is radial alternate exarch condition. Radial alternate exarch condition, but polyarch condition. Polyarch. But in dicot, maybe diarch or tetrarch or octarch, not more than 8. But in monocot, more than 8. And if you see the medulla, pith region also, you can see in diagram also. Here, the monocot root, the center occupied by pith. It is made up of the parenchyma cells. Either these are different in shape. This function of the medulla, the storage food. Mostly it is parenchymatous, but very rare is sclerenchymatous also. So this is about dicot root and monocot root. Right. Here also same given some differences between dicot root and monocot root, page number 59. No, if you see the differences, here the only differences in steel, remember, differences in steel. There is no differences out of the steel. That means no differences in the epidermis, almost similar. Cortex, no differences. That only one small differences in dicot cortex and monocot cortex. Here the dicot cortex comparatively narrow, but monocot cortex is some more wide, narrow in dicot, more in monocot. And another important difference, already told you, some secondary growth also takes place from the cortical cells or parenchyma cells, but the secondary growth not formed in the monocot root. Endodermis, both have the endodermis Casparian bonds, but Casparian bonds less thick in dicot, but very thick in monocot. That is one another difference. That is also small difference. Both have the Casparian, but the composition different. Right. Lateral roots. But pericycle mostly produces lateral roots, but pericycle not produce lateral roots in monocot. Same. And another most important thing only in xylem. Number only in steel, that is the inside the steel, the number of vascular bundle less in dicot root, more in monocot root, and medulla absent in dicot if it is present very less, and more medulla present in monocot root. So that is the difference between dicot and monocot root. And next one, dicot stem and monocot stem. Let's see the stem, dicot stem and monocot stem. Right. Page number 47 clearly given about the diagrammatically explain dicot stem and monocot stem. What is stuck here? Right. Right. So this is dicot stem and monocot stem. Right. First dicot stem. Like root, it also same epidermis, actually not given here, and because no differences in the root loop, almost similar. First epidermis, second cortex, steel. Three regions. Right. First epidermis, common, uniciliate, without intercellular space, cutinization present like a leaf, but root no cutin, no stomata. And here the if tender stem stomata also present, but not matured one. Right. The same carry forward to monocot stem also. The monocot stem also, epidermis uniseriate, single layer, without intercellular space, parenchyma, thick cuticle, and continuously maybe few stomata also may be present. And from the epidermis outer growth, trichomes are absent. That is one important. If the stem epidermis outer growth, trichomes present like this, these are the trichomes. But this type of this multicellular trichomes absent in monocot stem. That is an important. Hypodermis. That is next after epidermis, next one cortex. Here cortex clearly distinguished into three regions: hypodermis, hypo, normal cortex. Last one already you know, endodermis. Normal cortex and last one endodermis. Endodermis. Right. First, let's complete dicot. Hypodermis. Next to the epidermis, it is the first part of the cortex, and the stem of the sunflower. That means, of course, the sunflower is the dicot. This hypodermis made up of coloncyma cells. Coloncyma function give mechanical to the stem, mechanical strength to the stem. After hypodermis, parenchymatous normal cortex present. Almost in hypodermis without intercellular space, and which is made up of coloncyma tissue. Hypodermis made up of coloncyma. The function give mechanical strength. It is maybe 2 to 3 layers. Of course, the cortex part, it is 2 to 3 layers. Next, normal cortex more than 4 layers with intercellular space, and which is made up of parenchyma cells. And the innermost layer of the cortex, that is endodermis. This endodermis, it consists starch grain called starch sheath. The cells arranged without intercellular space, this limiting extracellular tissue cortex, same like a root system. After endodermis, the next one, steel region. But in endodermis, not clear in both stems. Of course, in monocot stem, completely absent. If you see hypodermis in monocot sclerenchyma. Only one difference is dicot hypodermis collenchyma, but in monocot hypodermis sclerenchyma. That is the difference. The same here or same mechanical strength. And next one, no clear differentiation into normal cortex and endodermis in monocot stem. They are together. That means that in place of normal cortex, that is called ground tissue. That is called ground tissue. No endodermis. Endodermis not found in monocot stem. If you take the steel region also, in steel, pericycle, medulla, vascular bundle. The pericycle this is the represented by the cell layer that occur between the starch sheath and vascular bundle. That means starch sheath, that means that is like endodermis. This is the limiting layer between the endodermis of bundle, endodermis of the starch layer and vascular bundle. It may be made up of sclerenchyma. sclerenchyma patches. It appeared sclerenchyma patches, but this pericycle absent in monocot stem. And next, another important vascular bundle. The vascular bundle arrangement in dicot as a ring. You can see in diagram as a ring, and type of vascular bundle different when compared with the root, both dicot monocot root. Here the vascular bundle conjoint collateral, and open type. That is important. Conjoint, collateral, open type. Open type means conjoint means both are joint. That means xylem, blue color. This is xylem, outside xylem. This one, xylem. The green color, this one, phloem. Inside phloem. And the xylem and phloem separated by meristematic tissue. It is separated by meristematic tissue. That's why it is called open type. If the meristematic tissue absent, that is closed type, like a leaf, like a root. A root that is closed type. But here the open type because of having the meristematic tissue between the xylem and phloem. And here also same xylem outside and phloem inside. And the protoxylem inside. So this condition called endark, whereas in root exarch condition. Let's see vascular bundle clear structure. For example, this is the vascular bundle. This one. This is one vascular bundle, and it is center separated by meristematic tissue. Sorry, meristematic tissue. Sorry, this right. It is reverse. The outside blue color phloem. Inside this one xylem. I wrongly explained. Sorry for this. Right. So phloem. Outside phloem. Inside xylem. The xylem that toward the medulla region. Xylem, first xylem. This type of development. So this type of development, this condition called as endark condition. This is protoxylem, this is metaxylem. So this condition called as endark condition. And still another part in dicot, medulla, the center medulla present. Medulla or pith, medulla pith present in dicot stem. Right? This is about dicot stem. Now the monocot stem, you can see diagram. There is no ring of vascular bundle. No ring. Is there any ring like dicot? No ring. But these vascular bundles scattered. These vascular bundles scattered in the ground tissue. Scattered in the ground tissue. That means this, for example, this is epidermis. Below the epidermis, 2 to 3 layers. Of course, from here, 2 to 3 layers. Hypodermis, sclerenchyma, hypodermis. After hypodermis, the rest of the place called as ground tissue. Whereas in dicot stem, clear normal cortex and last layer endodermis like part is there. But is there the entire portion called as ground tissue. No steel here, no pericycle, no endodermis, no steel, clear steel. But within the ground tissue, the vascular bundles arranged, scatteredly arranged. Like this, you can see this is one vascular bundle, this is one vascular bundle. And within the ear, the difference between the vascular bundle. So these are the scattered in the ground tissue and more crowded toward the periphery than the center. You can see in diagram also, center almost nil here, periphery more. So this type of steel called as atectosteel. This is very important. The scattered steel called as atectosteel. steel. Scattered one called as attractive steel. This round steel called as U steel. This round one called as U steel. This round one called as U steel. And this is attractive steel. Bundles conjoint same like dicot stem, collateral same in dicot stem, end arc same in dicot stem, but closed type. Here open type in dicot stem, but here closed type. That's why there is no meristematic tissue. That's why does not show the secondary growth. Of course, some monocots also show subnormal secondary growth. I will explain later in secondary growth class tomorrow. And there is no medulla in the center of the stem. So this is the difference between dicot stem and monocot stem. This is very already you must have learned during practical training classes about the stem, dicot stem, monocot stem, internal structure, and these are very important. And some distinguishing characters between the dicot stem and monocot stem. Let's see. First of all, dicot stem multicellular epidermal layer. Both first of all similar character as well as dissimilar character. First, both have the epidermis common. Both have the single layered without intercellular space. But in dicot from the epidermis, multicellular trichomes are developed, but these are absent in monocot. That's why you can see in our daily routine life, wherever the plants are available, you can see the surface of the dicot stem and surface of the monocot stem. Mostly the monocot surface smooth when compared with dicot. The dicot, there is a small airs are present. We can see directly our naked eye the airs. And the monocot stem no silica deposition. Sorry, dicot stem no silica composition, but is monocotyledon silica present. In next, the epidermis complete. In cortex, both have the first region hypodermis without intercellular space mostly without intercellular space, and but difference is the dicot hypodermis collonchyma, monocot hypodermis sclerenchyma. Here the remaining ground tissue differentiated, but in monocot not differentiated. Steel separate. That means the ground tissue separated steel and extrastellar tissue in dicot. Stellar and extrastellar tissue not separated in monocot. And another important, the dicot steel, already told you, U steel. U steel means around the medulla or pith, vascular bundle arranged in a ring. But in monocot, that is atectosteel type. Atectosteel type means within the steel, of course, the ground tissue itself is considered as a steel. In the steel, the vascular bundles are scattered, arranged in scattered manner. That is atectosteel. Vascular bundles are limited in a ring, limited, maybe 8, 10, but in monocot, vascular bundle unlimited, more than 20. Another important bundle type, conjoint, collateral, open. The same here also monocot, conjoint, collateral, but not open, closed. Already told you why it is closed. And in dicot, protoxylem lacuna, that means in clear space, absent. Protoxylem lacuna present in monocot stem. Protoxylem lacuna absent in dicot, present in monocot. Bundles are with the bundle cap, clear cap is present in dicot, but there is no cap in monocot. A vascular bundle does not consist any sheath. But in monocot stem have the sheath, present the sheath with normally present but normally absent in monocot. So these are the some important points of the dicot stem and monocot stem. Clear? Now another important, already this internal structure of the root, internal structure of the stem, you must have learnt in during practical training classes with experimentally. You can take a section, you can take a units and make a section, and you must have observed under microscope. Clearly, no doubt. You can easily write about this theory. But abnormal characters, abnormal secondary growth, very, very important, and some more difficult when compared with normal secondary growth. So normal internal structure. Now let's next one, secondary growth. Another important topic. It's very, very important topic, secondary growth, normal secondary growth and abnormal secondary growth. Right. First of all, what is primary growth? What is secondary growth? Let's see here. What is primary growth and what is secondary growth? Primary growth, which is happen with help of apical meristem. Due to the apical meristem have the capable of division, due to the capable of division, it is always shows upright direction. Apical growth. That is primary growth. The linear growth, that is called primary growth, while the plant body goes upright direction and automatically apart from the upper growth, the branches also arised, developed. So that is the not possible. The if it is thin stem present, weak stem present, it is not possible to bear that such a branch. That's why the need to improve that stem, the weak stem need to enlarge, need to improve and shows the radial growth. The radial growth of the stem called as secondary growth. Here, if we take the linear growth, upright direction growth, linear, that is primary growth, which is takes place with help of the apical meristem, and radial growth, width, 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 that is called secondary growth. Right. I think clear definition clear. Primary growth and secondary growth. I already told you the primary growth takes place by the apical meristem. Then how the secondary growth takes place? The secondary growth takes place with help of meristematic tissue, which is present in vascular bundle. The vascular bundle, meristematic tissue, and between the vascular bundle meristematic tissue all together form a ring. After formation ring, the inside and outside tissues are formed. Due to the formation inside and outside, what happened? The tissue size increased. When the tissues increase, automatically the pressure falls on epidermis. Epidermis enlarge in size, and it triggers to the radial growth. This is normal secondary growth, and which is happening in steel between the vascular bundle. And abnormal secondary growth. And the second one, but this is not completed. Secondary growth not completed. Just give the introduction and definition. Abnormal secondary growth. It is completely opposite to the normal. A normal steel vascular bundle. And also in normal, sometimes it is involved in cortex cells also in simultaneously. Cortex vascular bundle formed while the growth occurs to withstand the epidermis, and the pressure of the steel. The cortex regionals need to improve. That's why this is also part of the normal cortex. I will explain clearly diagrammatically in tomorrow class. Whereas in abnormal secondary growth, the secondary growth may be formed by pericycle cells, by medulla cells, without vascular bundle activity. And also the abnormal activity not formed in cambial ring, and also presence of the vascular bundle also. Actually, vascular bundle present around the steel, inside the around the medulla, inside the steel. In abnormal situation, sometimes this vascular bundle may be present in cortex, may be present in medulla. This is abnormal activity. So this is abnormal characters. I will explain later tomorrow. Secondary growth, normal and abnormal. And this is secondary growth and abnormal secondary growth, very, very important in theory exams. Right. Thank you all.